All right, welcome everybody. Happy Friday afternoon. That's always my favorite thing, Friday afternoon. So I'm going to present a little bit about the research that goes on in my lab, but I'm really just an opening act. The main event, of course, is Manny Flores, who is a grad student working in the lab. So I'm hopefully not going to take too much of his time. He has permission to throw things at me to get me to shut up, because professors tend to not shut up. So what I want to highlight first, though, is not me, it's my students. So the students who work in the lab are really the people who drive all the projects forward. I mostly sit in my lab office, and I'm kind of cranky, and they do everything. So I sit there, and I get a lot of credit for it, which is kind of silly because they're the ones who do all the work. Two of the students that I have co-mentored with other faculty are defending or presenting their master's thesis work. So if you're around sometime next week after Thanksgiving, we've got a couple of master's thesis presentations. I've also had a number of students graduate from my lab and move on to bigger and better things, and I'm so proud of all of them as well, especially the papers that my students have published. So these are all papers that have come out of the lab that are authored or co-authored by students who've worked in my lab. So who am I? In order of my daily thought process, I usually think about cats, because there's usually one that's sleeping on me. I think about dachshunds, because I have to let them out to go to the bathroom. And then in no particular order, I think about coffee, birds, and bacteria. Those of you who take my 8 a.m. microbiology class, you know how important coffee is. Those of you who work in my lab know how important birds and bacteria are. So it really depends on the day how those come out. I have a bachelor's degree in biology with a minor in chemistry and a concentration in genetics from Cal State Stanislaus, so just up the road. A master's degree in biology from University of the Pacific. A PhD in cell and molecular biology from UT San Antonio. So if I say y'all, it's because I lived in Texas for 8 years doing a PhD and a postdoc. So but I am from California. I did my postdoc with the U.S. Army Institute for Surgical Research in the Dental and Trauma Research Detachment at Joint Base San Antonio, Fort Sam Houston, which we call USA ISR DTRD JBSA Fort Sam Houston. You have to say it all in one breath or it doesn't count. And I studied antibiotic resistance. My teaching responsibilities are mainly micro, med micro, genetics, bioinformatics, and then because I did like birds, see birds, I also teach classes like field ornithology whenever I get a bug and decide to do something fun with Dr. Constable. Okay, what goes on in the lab? We have a number of different projects in the lab. Manny's going to talk about probiotics, but we have projects looking at things like the crow fecal microbiome. So we do get to do sometimes a little bit of field work. And the crows come streaming in. They come roosting in the winter. So this was a project initially started by a former grad student, Lee Nelson. We're doing a microbiome of Scout Island in conjunction with one of the chemistry courses. We study bush tits, which are these cute little birds on campus, their nests, and their microbiome, which this is me in a cherry picker in a eucalyptus tree on campus. I'm super afraid of heights, so that wasn't fun. But field work, you do what you have to do. We also study bacteria like Pseudomonas aeruginosa and Klebsiella pneumoniae and their antibiotic resistance. So this is Pseudomonas aeruginosa in culture. It produces this really cool blue-green pigment. When patients have an infection with this, you can actually see that their wounds turn that color. So you know that they have that infection. And finally, we study tick-borne diseases in Fresno County. And so this is a tick trap that one of the students designed, and you can see the tick, some of the victims, right? Some of our victims already. But then another tick crawling onto the tape. So these are just the projects that are mostly in my lab. This doesn't have anything to do with all of the collaborations that we have with labs outside of the department, outside of the college as well. So we do a lot of work mostly focused on bacteria. And what we're going to talk about today really is antibiotic resistance. And so why do we talk about resistance? Resistance is futile, right? If you're a Star Trek fan, so if you're trying not to be assimilated, you don't have a choice. You're going to be assimilated. But if you look at it from the perspective of the bacterium, is resistance a futile strategy? No, it's not. In fact, in late 2016, we had our first patient in the US with a Klebsiella pneumoniae infection that was resistant to all 26 FDA-approved antibiotics. They call it pan-drug resistance, resistant to every single antibiotic. And that patient did die because there was no therapy to treat her. So these are our super bugs. If you were here last week, Dr. Wazlakov talked about her super weeds. These are super bugs. In the United States, antibiotic resistance is a huge public health concern. The CDC published a report back in 2013 on their antibiotic resistance threats. And so we've been using those numbers for a really long time. And then fortuitously, because somebody had to smile on me at one point or another, the CDC released a new report about a couple of weeks ago with their updated numbers. So their updated numbers are that in the US every year, when you combine drug-resistant bacteria and fungal infections with Clostridioides difficile, sometimes people call it Clostridium difficile, but the taxonomy is Clostridioides now. When you combine these, over 3 million people in the US every year will develop an antibiotic-resistant infection. And you add up here, nearly 50,000 people a year in the United States die of these infections. So it is a major public health concern. But not only in the US. Worldwide, the UN Ad Hoc Interagency Coordinating Group on Antimicrobial Resistance released a report earlier this year that worldwide 700,000 people die of drug-resistant infections every year. And about 250,000 people die of multi-drug-resistant tuberculosis alone. So this is a huge current public health threat, and unfortunately it's not getting much better because this same group estimated that with increased hospital costs, with increased lack of time at work, that by 2030, drug resistance will force 24 million people into what they consider extreme poverty because you can't afford your medical bills anymore, because you can't go to work. And they estimate that by 2050, 10 million people every year will die of drug-resistant infections worldwide. That's more than cancer. So this is a major global public health threat. And what we see is that, first of all, there are no new drugs really being put onto the market. Most major pharmaceutical companies have completely shuttered their antibiotic research and development. One, if you think about, I could spend $1 billion in 20 years to develop a new antibiotic that a patient might take for 10 to 14 days. That doesn't sound very lucrative because you could spend the same investment, $1 billion in 20 years, to develop a drug like Lipitor to lower cholesterol, or Viagra that somebody will take for the rest of their lives. Those two drugs alone generated more than $20 billion a year in profits for Pfizer. So who's going to fuss with antibiotics? But let's say they were going to be altruistic and do it anyway because we need it, and they're nice, the nice drug companies. The microorganisms make it very difficult to want to design new drugs. Every time an antibiotic is introduced to the market, we see drug resistance emerge. And sometimes it's slow. Penicillin was introduced in 1943. The first penicillin resistance was in 1967 in a patient. 24 years, that's pretty good. That would have been a decent investment. Okay, well, here's methicillin, introduced in 1960. Methicillin-resistant Staph aureus, 1960. So the same year we introduced a drug into the market, we already identified resistant bacteria. Here's one I really like. Fluconazole was FDA approved in 1990. Fluconazole is an antifungal. But we already had fluconazole-resistant Candida albicans, which is a fungus, in 1988. So 2 years before the drug even got approved, we already had resistance circulating in patient populations. And then you get really fun things like this, the introduction of daptomycin in 2003, and the emergence of daptomycin-resistant, methicillin-resistant Staph aureus in 2004. When we introduce a drug to market, it's not a question of if microorganisms will evolve resistance to this drug. It is a question of when or how quickly. And so what we see, I'm going to show you a little video of what happens in a laboratory setting. And so this group at Harvard has created a mega plate. It's 2 feet by 4 feet. Those of you who have taken microbiology, you work with tiny little 100 centimeter plates, 100 millimeter plates. And so what they have are different zones. On the far zone, they have no antibiotics. In the next zone in, they have the level of antibiotics that is going to kill the bacteria, the minimum level. Then they have 10 times that amount, 100 times that amount, and 1,000 times the amount that kills the bacteria. They put a thin agar layer mixed with these bacteria, and they put them on the plate to see what happens. So you see the white is the growth of the bacteria. They reach the border. Here are the antibiotics. They are stopped. That level of drug kills them until it doesn't. So they started to spread on this side, and now they started to spread on this side. And you'll see they'll hit a little wall again, and they'll slow down, right? They kind of hit a wall, and then they don't. They breached the wall. Now they're growing in 10 times the amount of antibiotic that should have killed them. And then they hit another wall, and now they're growing in 100 times the amount of antibiotic that should have killed them. And pretty soon, they'll hit that final wall, and now they're growing in 1,000 times the amount of antibiotic that should have killed them. This experiment took 11 days. So in 11 days, these bacteria were able to grow in 1,000 times the amount of antibiotic that should have killed them previously. When you take antibiotics for 10 to 14 days, what do you think you're doing inside of your body? It's like Lysol, kills 99.9% of germs. Ooh, that 0.1% though. Okay? So when you take antibiotics, you're basically showing them these are our weapons. And you will never, ever be able to grow as fast as they can. If you think about human generation time, right, 25, 30 years versus E. coli generation time, 20 or 30 minutes, you're never going to win. So what are these bacteria actually doing that is allowing them to be resistant to these drugs? Most likely what we see on that plate is a collection of successive mutations that have changed the antibiotic's drug target. This is very, very common. If you change a single nucleotide, you have the potential to change a single amino acid. And if you change that single amino acid, you have the potential to change the shape of the protein. And if you change that protein shape in such a way that it can still function, but it no longer binds the antibiotic, you now have an antibiotic-resistant bacterium. Other mechanisms for antibiotic resistance include the use of efflux pumps. Drug comes in, drug goes out, drug comes in, drug goes out. So theoretically, the antibiotic should still work, but it never can make it to its target because the bacterium just throws it back out the window. You can close your doors and windows and restrict access to antibiotics. You can change or destroy your antibiotics with the development of new enzyme pathways. This is most common with penicillin resistance. So penicillin and the derivatives are a class of drugs called beta-lactams. They have a specific ring structure. Many organisms have an enzyme called a beta-lactamase. It's basically a pair of scissors that says, oh, look, a beta-lactam, and it inactivates the drug. And then finally, bacteria and fungi can develop new metabolic pathways to bypass the drug target. So the drug still gets in, the drug still binds its target, but the bacterium has a backup mechanism to do the same function. And this is what we see with methicillin resistance. The methicillin-resistant Staph aureus have a brand new enzyme that helps them build their cell wall. So when they are exposed to methicillin, they turn on this brand new enzyme. They allow methicillin in. Methicillin binds its original target, but Staph doesn't care because it has a whole new pathway to do the same thing. So that's scary in and of itself. Microbes will always evolve faster than us because they replicate faster than us. They get to numbers more than ours, more than we could even imagine. So overnight in here, you would have trillions and trillions of bacteria, all of them with different mutations. Hopefully not, because I won't want to drink that tomorrow morning if it has trillions of bacteria in it, but theoretically it could. For me, one of the scariest things is not that they can evolve resistance, but then they go ahead and share it. So they make friends with closely related or distantly related microorganisms, and they can share mechanisms for antibiotic resistance. They can use plasmids or extra chromosomal pieces of DNA. They can use transposons, or sometimes called jumping genes, that can insert themselves in and out of the chromosome. And they can also use phages or bacterial viruses to move antibiotic resistance around. And so here's our 3 common processes if you've taken micro. Hopefully this is a review. If you're currently in micro, this will probably be on the final. Hint, hint, I see a bunch of you in here. Okay. Transduction, using viruses to move gene elements, right? Conjugation, directly sharing and having this pipeline where I give you something. And transformation, the ability to pick up naked DNA from the environment. So all of these things that allow bacteria to pick up DNA from completely unrelated species also allow them to transfer antibiotic resistance. And so I told you this is one of these emerging global health concerns. Why didn't anybody warn us? Because I bet all of us at one point or another took antibiotics when we didn't need to. Right? Any new parent, their kid has a sniffle, you run them to the pediatrician, give them antibiotic. Okay. Right? They probably have a cold. Antibiotics don't work against viruses. But these drugs were prescribed because it made people feel better. Drugs are used to help animals grow bigger. Drugs are used inappropriately. And so then we have this constant exposure to antibiotics, this constant learning of our techniques to fight them. And they learn back. So why didn't anybody tell us that this was going to be a problem? Why didn't we know? Well, we did, we just didn't listen. So Alexander Fleming was, of course, the discoverer of penicillin. And in his Nobel Prize speech in 1945, he gave an example of a couple where the husband had an infection. He was prescribed penicillin. He took it until he felt better, and then stopped taking it. And then his wife developed the same infection, so the husband said, well, here, take the rest of my prescription. But she ended up getting a penicillin-resistant strain of that infection because he didn't completely eliminate his with the antibiotics. And so he said in his speech, if you use penicillin, use enough. Use what you should. Don't stop taking it just because you feel better, because you've got those cells in there that have learned and will come back and get you. So you hold on. The human didn't take the full course of antibiotics. You're going to make it. We shall rise again. So then the question is, what does this mean without the ability to use antibiotics? So they call it a post-antibiotic era. And they say basically that means an end to modern medicine as we know it. Things as common as strep throat or a child's scratched knee could once again kill. And so I would argue that the question is not whether are we approaching a post-antibiotic era, but it's what do we do now that we are in one. Every 15 minutes, someone in the US dies of a drug-resistant infection. So what do we do? Okay, so in part, the students in my lab and I are interested in trying to figure out what we can do. So these are some of the pathogens that we study in the lab: multi-drug resistant Pseudomonas aeruginosa, methicillin resistant Staph aureus, extended spectrum beta-lactamase producing Enterobacteriaceae. That's a fancy way of saying multi-drug resistant bacteria like Klebsiella pneumoniae. And we don't quite bring this one in yet because it's too scary even for me. It is threat level urgent, highest level of concern by the CDC, carbapenem resistant Enterobacteriaceae, basically pan-drug resistant bacterial infections. Combined, the microbes we study cause over 500,000 infections in the US every year, 23,500 deaths, and costs of $3.8 billion per year just in the US. So what do? Well, students who take Bio 120 are doing new drug discovery. They look for microbes in the soil that produce novel antibiotics. Students in my lab are working on identifying genes that, when mutated, lead to antibiotic tolerance or antibiotic resistance. Students in 120 are also looking for phage, right? Bacterial viruses that could potentially kill some of these drug-resistant bacteria, so we no longer would be reliant on antibiotics. We're looking into vaccine development. We have generated knockouts in Klebsiella that are avirulent in an insect model. What happens when we use those as potential vaccine strains? And finally, let's look at the synergistic effects. One antibiotic no longer works. What if we put together two? Right? Can we make that be something that, at least as a Band-Aid, can get us through? So I thought I would leave you on that kind of optimistic note. We have things that we can do. But then I thought, no, that's no fun. Let's go ahead and leave you with a negative note. And that's where I'm going to introduce Manny. So Manny is a grad student in the lab. He joined my lab back in 2017. He got his bachelor's degree here. He's in his second year now as a master's student. He's a Bridges to Doctorate scholar. His research in probiotics and antibiotic resistance has been featured in national news. He was selected for a rapid-fire talk at our national meeting, which has over 11,000 attendees. Right? He gave his poster presentation. Right? He got his bachelor's with us. So I don't want to steal any more of his thunder. I'd like to introduce Manny Flores to present his master's thesis project.
Hello, my name is Manny, and what I'm going to be talking about is antimicrobial resistance in probiotics. So I guess a little bit more detail is my thesis covers Bacillus strains found in these probiotics, and then some work I've done over the summer at UC Merced with our collaborator for my thesis focuses on multistrain probiotic dietary supplements. And so we'll be studying antimicrobial resistance in those. So probiotics are live microorganisms that are typically bacteria or yeast that confer some type of health benefit to their host that consumes them. And they're claimed to help replenish the microbiota for whatever application you use them for. And they do that through competitive exclusion, so they'll basically kick out a foreign microorganism out of that normal microflora and then maintain it back to its normal state. And they're also claimed to have helped boost neurological, immunological, and endocrinological systems of humans, pets, and commercial livestock. And commercially, they're available in monostrain or multi-strain form. So monostrain just has like one particular strain stated on the nutritional label, whereas a multi-strain form will carry more than one strain of bacteria. And usually the multi-strain will have like different species. And so probiotics in general, they come in various forms that we can buy just anywhere. So they'll come in dog food, kefir shots, a probiotic version of root beer, Japanese fermented soybeans, kombucha, cannabis chocolate, deodorant for your armpits, enemas, and then dietary supplements. And so this is the mechanism of action. So just imagine you're eating a probiotic taco. And so you're going to eat it, and then basically it's going to be digested, and it's going to release spores in the lower parts of the gastrointestinal tract, where it's going to colonize either temporarily or permanently in the gut, and then kick out some foreign bacteria that was pictured in that red oval. And so in the gut, you have sort of like a paradise for the exchange of those mobile genetic elements like plasmids, and typically those will confer antimicrobial, or those will carry genes that confer antimicrobial resistance. And so you have commonly an exchange of antibiotic resistance between one bacteria to another in the gut. And so in that context, the UN recently released a report stating that 10 million people annually, or 10 million people would end up dying from antibiotic-resistant infections annually by the year 2050, alongside with global expenditures of about $100 trillion in the developed world. And that sounds like a Dr. Evil level amount of money, but that's just the reality of the emerging risk of antibiotic-resistant infections. And so one way to mitigate this emerging risk is through monitoring of antibiotic resistance. So what we're doing is basically monitoring the transfer or the exchange of antibiotic-resistant bacteria between probiotics and pathogens of the gut. So our objectives for my thesis, so our target bacteria is Bacillus, and we want to analyze, or we first want to determine antibiotic resistance profiles. So we want to find out which antibiotics they're resistant to, and we want to also analyze our potential to transfer that antibiotic resistance to harmful bacteria. And then later on, I'll be talking about the multistrain probiotics. So our objectives for those were to determine the taxonomic the composition of the multistrain probiotic supplements. And then we also analyze the presence of chromosomal and mobile antibiotic resistance genes. And then from there, we then did some bioinformatics analyses where we assembled genomes out of raw sequencing reads and visualized them. So Bacillus probiotics, so this is like what my thesis is about. So Bacillus, they are ubiquitous bacteria, so they're found everywhere in the environment. They are gram-positive. They form spores, and thus they display the vegetative and endospore cell morphologies, pictured right here. So this is a gram stain, that's the endospore stain, showing these green little dots everywhere, the spore morphologies. And in the probiotic manufacturing process, these spores are very valuable since these spores are able to withstand the harsh environments of mass production of these probiotics, and they're able to remain viable for years, so they're pretty reliable in that process. And they're commonly used as probiotics. So methods involve collecting our samples. So what we did is we did some field work, and we literally went out to like Whole Foods, Target, Googled stuff on the internet to collect our samples. And so we just bring those products into the lab, and then we heat those samples up to 80 degrees Celsius, so that'll just isolate the Bacillus endospores. And from there, we can just start cultures from those spores. So what we did is we isolated the spores and did gram and endospore staining to have a tentative verification of Bacillus in our sample. And then we did herbivour assays to develop antimicrobial resistance profiles. So it's kind of like a Tinder profile of like their likes and dislikes. So we know which antibiotics they like and which ones they dislike. And we did that not only for the probiotic, but also for the pathogen. So far I've done that for Salmonella enterica. And that's going to be helpful when I explain later. And then we've done whole genome extractions, and we plan to do whole genome sequencing either in-house or at off-site facility. And then depending on how we do it, or however we do it, is we're going to do whole genome and plasmid assembly through a program called SPADES, which I'll go over later. And then we can visualize those assemblies through a program called Bandage. And we can also analyze their inherent or their mobile antibiotic resistance through the Comprehensive Antibiotic Resistance Database. And there we'll just align our sequences to those on this database that contains nucleotide sequences conferring antibiotic resistance. And we're going to look for homologies in those sequences compared to ours. And then that's the min-ion sequencer that I was referring to up there. So for the Kirby-Bauer assays, on the far right, we have a plate that I just swab a whole culture of a probiotic on. And I then plate a panel of 15 antibiotics against that culture. And what happens is that disc has a minimum inhibitory concentration of an antibiotic. And that antibiotic overnight should develop a zone of inhibition around the antibiotic disc. So areas where you see no bacterial growth on the plate, that's the zone of inhibition. And so I measure the diameters across that zone of inhibition. And across each trials, I get an average. And then I get those averages and then compare them to the Staphylococcus zone interpretive standards, which are used to determine whether or not you have resistant bacteria, intermediate sensitivity to the antibiotic of your culture, and then susceptibility to the antibiotic. So anything in the resistant value, if it's below or equal, would be resistant. If it's intermediate in sensitivity, it would fall within that middle range. And then on the right, if it's equal to or above, that's susceptible to that antibiotic. And then we did that across a panel of 15 antibiotics. So our results so let me actually explain the graph a little bit. So on the x-axis, you have the panel of antibiotics, and on the y-axis, you have the average zone of inhibition. In this case, for the Bacillus coagulans strain, so that's isolated from a dietary supplement. And so you have in the dots the average zone of inhibition that they produce against a particular antibiotic, and in those colored bars, so in the gray, you have the resistance ranges, and in the orange, you have the intermediate ranges, and then anything above that in the white would be susceptible. So for this particular strain, we have ampicillin. We have observed an average zone of inhibition within the resistant ranges for ampicillin. And then we have it for bacitracin. So one thing about bacitracin is that this is actually naturally produced by Bacillus species. So this kind of works in our favor as a positive control. So there's that. And then we have observed zones of inhibition in the intermediate sensitivity range for clindamycin and rifampin. And then for Bacillus coagulans MTCC5856, isolated from target brand kombucha, we have the average zone of inhibition falling within the resistant range for ampicillin, and then bacitracin, and then intermediate sensitivity for clindamycin and rifampin again. And then Bacillus coagulans GBI30 showed, again, ampicillin resistance, and then bacitracin, which is expected, and then susceptibility to the rest of the panel. And then Bacillus coagulans IS2, this was isolated from a probiotic gummy. Again, bacitracin resistant, and then had intermediate sensitivity to meropenem. And then Bacillus subtilis, a non-GMO strain, which is like how it's labeled on that product, showed average zones of inhibition for ampicillin in the resistance range. Again, for bacitracin, clindamycin, and erythromycin, and it had intermediate sensitivity to chloramphenicol and ceftriaxone. And since we haven't gotten quite to the whole genome sequencing yet, we weren't able to do card results yet for our strains, but on GenBank we were able to download whole genomes of Bacillus coagulans and Bacillus subtilis species, totaling to about like 150 total. And we found genes conferring resistance to several antibiotics. So this is that column right here, the genes, the gene family right here, the drug classes that they confer resistance to, and the resistance mechanisms in which they function. So for the next part, we've done conjugation. So we facilitated the exchange of antibiotic resistance genes between the probiotic and bacterial cultures, and then we do conjugation assays to achieve that. So basically that just involves growing cultures of our probiotic and our pathogen to mid-log phase, which is a phase in which they're most likely to exchange genetic material. So we grow those separately, and then we mix them and then incubate them together overnight. And then what I've done so far is I've worked with Bacillus subtilis, a non-GMO one, and Salmonella enterica. So Salmonella enterica is gram-negative bacteria, and so to filter out the Salmonella from the Bacillus, I would then plate that mixture onto selected media, such as McConkey, which selects for the growth of gram-negative bacteria. So it'll only grow the Salmonella and not the Bacillus. And so when I plate it, I just do a streak for isolation, and I get 3 colonies, and then I am able to run 3 trials of post-conjugation Curry-Bauer assays. And then depending on our results, I will do whole genome extractions and then do whole genome sequencing, depending if there's a suspicion that there was a transfer of antibiotic resistance from the probiotic to the pathogen. And so this is pre-conjugation results. So I did Curry-Bauer assays on the Salmonella strain, and these are the resistance ranges right here. And this is the intermediate sensitivity range for Salmonella. So before conjugation, I had an average zone of inhibition within the intermediate sensitivity range. And then after conjugation, that average fell within the resistant ranges. And that was towards ampicillin. So before conjugation, this is what it showed for ampicillin, or against ampicillin, and then after I did the conjugation, I then plated it against, I plated the ampicillin against the Salmonella after conjugation, and this is what I got as far as the average zone of inhibition. So I don't have the other results that I've had visualized yet, but 2 other species of Bacillus, so Bacillus coagulans and Bacillus coagulans perdura and the GBI30, the 2 different strains. I did conjugation with those, and then those appeared to have increased the susceptibility of Salmonella enterica to ampicillin. So it's kind of it's dichotomous so far as my results go. So I'm going to go into the multistrain probiotics that I've researched over the summer at UC Merced. So basically, I was given raw sequencing data. So that raw sequencing data comes in FASTQ files, which I can then input into various bioinformatics tools that are available free online. And the raw sequencing data comes from samples of multistrain probiotics, such as Dr. Tobias Deep Immune, Vitamin Bounty Pro, a Once Daily Woman's, Flores, and then the InfoWars Biome Defense. And so this is my workflow, is I got the sequencing files, and then I was able to do a quality control to see whether that sequencing data was good enough to do downstream analysis. And then upon those results, I then did de novo genome and plasmid assembly, and I did that through a program called SPADES and Plasmid SPADES. Then I did taxonomic classification of the probiotic supplements, then analyze the presence of antimicrobial resistance genes, and then visualize these genome and plasmid assemblies through de Bruijn graphs. So those are colloquially known as spaghetti graphs, and you'll see why. So I use a program called FastQC for quality control, and that just has different modules that tell you whether you have good per base sequencing data, your sequence like distributions are good, and then your per sequence GC content is good, for example. So this is a good quality score data right here. So you want a score above 28 across your bases. And then you want a distribution of your sequence links. Basically, you want it to be skewed over to this side and then up high. So that basically is the score right here. And then you want your GC distribution to basically fall within this blue line. So that's a theoretical distribution of your GC bases. And then next I did the plasmid, or the genome and plasmid assembly through spades. And the spades assembler works in different stages. So basically it just assembles your genomes of plasmids by constructing de Bruijn graphs and then estimating how long your strings of DNA are. And then from there, it'll just start connecting those strings of DNA to one another. And so basically what it does is that it forms contigs, which are basically, they're strings of DNA just put together, and then it'll form scaffolds, which are basically contigs put together. So that's sort of like a hierarchy in which it'll assemble your genomes or plasmids. And then it'll assemble the plasmids almost in the same manner, but it'll just remove any like edges in the DNA that aren't connected to one another, and it'll just construct a plasmid out of those disconnected sequences. And so once I have assembled everything, I then did taxonomic classification. So what I did is I just got my sequences and aligned them to a database called RevSeq that has basically the bacterial genomes inputted within it. And then I ran greedy mode that just sets the basically the significance of your matches. And so I got greedy, and then this is what I got. So this is for the PR, so I think this is a Vitamin Bounty Pro. Basically, it's only going to work on the genus level. So you have the Phytobacterium, Lactobacillus, Lactococcus, Streptococcus, which are basically, as it's labeled, are probiotics, or are genera that have probiotic species within it. And then you have unclassified, whatever it is, we don't know what that is that's in there, it just says unclassified within the probiotic. And then for the InfoWar sample, across 6 sequencing trials, we found Bacillus, Bifidobacterium, Lactobacillus, and other genera that would have probiotic species within them. We also found stuff that, according to this label, aren't in the actual sample. So Clostridium, Clostridioides. It had Streptococcus, but not the right species. So I'll go into that later on. They also had stuff that we don't know what's in there. And then we analyzed the AMR genes through the CARD database. And then I'll go into that later on to show you kind of how I work with that and the genome visualizations. And those were done through the bioinformatics application for navigating de novo assembly graphs easily, or Bandage. And you can just load whatever you assembled on spades, and then you just visualize your assemblies through the spaghetti graphs. So I need to hurry. So these are the spaghetti graphs. So I can construct databases here, and I can just input nucleotide sequences that I found. So I can put the FASTQ files within this database and then get matches. And then I found in one of the InfoWar samples that I found RPOC, which confers the resistance to daptomycin. And so I did that across all the significant matches that I got on the CAR database. And then in the InfoWare sample, I found in the genomes, genes conferring resistance to various antibiotics, and then the species that I was able to match against through blasts. So they had Bacillus thuringiensis, containing a gene conferring antibiotic resistance here, and then you had Streptococcus pneumonia right here, which causes pneumonia, containing ERMB, which is conferring resistance to antibiotic. And then in the plasmid, we had the same matches, except for TET-W. And then for the once daily women's, which promotes immune health, digestive health, and then vaginal health in women, had just plasmids that confer resistance to various antibiotics, and then they were all tied to the Bifidobacterium animalis. And then this is a Flores genome, and again, it just had genes in the genome, and then in the plasmid, conferring resistance, and then they were all Bifidobacterium species within that sample that we found. So basically, the takeaway from all this is, as an alternative to antibiotics, people have, started kind of adopting probiotics as sort of like a way to alleviate malaise in the gut. And I guess it's good to know that they're beneficial, but is it a viable alternative to antibiotics if they are able to transfer resistance and if they carry antibiotic resistance genes? So is it a viable alternative? And then lastly, I guess with that same question, is it also viable if that probiotic bacteria can release toxins in the blood and kill us? So this is sort of the dichotomy that I was presenting. And then those are my references and my acknowledgements. So Dr. Van Laar, thank you for having me in the lab. Dr. Mars Sistrom, our collaborator, Dr. Leah Mujic for being on my committee. And then Bo Hayes, Rick Cullen, Aveline Brar, Alexander Hugh, and the rest of my lab mates for helping me out. The Bridges to Doctorate program, I forgot to put the grant number. The GNI Graduate Research Grant, and the DRGS Gracchus Award, and the Fresno Biology Department. Thank you for having me. And Eastern Merced for also having me over the summer. And then I'll take questions. So that's Alex Granville.
So any questions for Manny? You can stay here and look at that picture.
Yeah, I'll leave it up.
So questions? Uh-oh.
With that, I just talked about this earlier, but we would do RNA extractions, and then we do a qRT-PCR, and then we can get resistance profiles to see what they're expressing.
Yeah, Manny actually had his committee meeting this morning
Yeah.
too, so he's had a big day. Any other questions for Manny?
Yes.
Your mechanism of transfer of resistance genes, we talk about one configuration.
Yes.
So what is one is resistant and another is not?
Yes. Sorry, did I interrupt?
Yes, so
Yeah, so
was it two different species?
it's two different species.
Yeah, and how
So basically, yeah, so that's one of the things is we suspect is that ampicillin resistance that we saw was on the local plasmid that was then conjugated to salmonella or some pattern. We'd have to do more analysis to see. So the whole genome sequencing helps out, and also checking to see whether it was something else that was causing that.
So if you want to leave, you can, but if you want to stay and ask questions, that's fine too. We'll be up here and you can come and ask us some questions.
Thank you.